Hello, ladies and gentlemen of Mr. Hescock's Life Science classes. Coming back to you, we're going to finish the second part of Chapter 5, Section 1. So we did the structure of DNA and DNA replication. Today we're going to use DNA, so we got to do transcription and translation. The warm-up question there is, what is the subunit that makes up a strand of DNA? And that subunit is called a nucleotide. So to make a protein, you need three types of RNA. You have messenger RNA, which is abbreviated as mRNA, ribosomal RNA, which is rRNA, and transfer RNA, which is tRNA. RNA, as you see over here in this picture, is a single-stranded molecule. RNA does not have the T's. So here's a T. You will not find it in the RNA. You only find these U's over here. And RNA is made of a different type of sugar that we call ribose sugar. It's a little bit different shaped, and those are the three main differences between DNA and RNA. So DNA has double-stranded, RNA is a single strand. You get no T's, and you get the differences. You do get a uracil. Transcription, script means to write, is to copy down the information of the DNA into an mRNA strand. So you unzip the gene, the nucleotides of the mRNA match up to the DNA strand, A to U, C to G, and then we follow that along till you complement the genes. And I'll show you that here in a minute. So once you've copied it into an mRNA strand, that is now transferred to a ribosome. And the ribosome has two pieces, a large subunit and a small subunit. And they attach. And then this ribosome will allow, there's three bonding sites. You have these tRNAs bring in the amino acids, and they match up. So when we read the code or translate the code, that's translation. Translation is the actual building of the proteins. And these amino acids will get linked together and linked together and linked together, and that will grow into our protein. So we need a DNA strand first. So if I go C, A, G, T, A, C, C, A, T, T, A, A. So this is my DNA strand. This would stay in the nucleus. This is like where our gene is. My mRNA strand will form during transcription. So C, G, A would normally go to T, but it's uracil. So U, G, C, T, A, U, C, G, C, G, A, U, T, A, and then U, U. So this is my mRNA strand. Now my mRNA strand goes to my ribosome, and the ribosome will attach in these two pieces here. And they kind of sandwich around this. Then the next thing that happened are these tRNAs are going to come in. And these tRNAs have the anticodon. So if this is G, you'll get a C, A, G. And this tRNA is carrying an amino acid. And another one will come in. So it becomes A, becomes U, A, C. Here's another amino acid. And then they get attached. And a third one will start to come in, C, U, U, and here's my next tRNA. And it will be the next one to move into the complex. So literally what happens is we get this chain. Of amino acids. And this, from here to there, is translation. Here's what it would look like on a GIF I found online, just moving the pieces in, matching up. This is how Google draws it, where you have your growing amino acids from where the tRNA falls out the one side, the new one comes in on the other. All right, ladies and gentlemen, so that takes us to the end of DNA. We did DNA replication, where you use one half of the DNA to make a copy of the exact same thing. We have DNA transcription, where you take the code of the DNA, copy it into an mRNA strand so that it can be traveled to the ribosome, and then you translate it, which is translation, where we take the code and build the protein that it does. I'm Mr. Hescox. Until next time, make sure you get your work done, and I'll be seeing you soon. Be safe, be good. See you later.